Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1958 witnessed the formal publication of a succinct, yet epoch-defining paper entitled Independent Functions of Viral Protein and Nucleic Acid in Growth of Bacteriophage, authored by Alfred Hershey and Martha Chase. This deceptively straightforward title belied a conceptual revolution, marking a watershed moment in our understanding of molecular biology and genetics. The Hershey-Chase experiment, as it became universally known, provided compelling evidence that deoxyribonucleic acid, or DNA, rather than protein, constitutes the genetic material responsible for directing viral replication. To fully appreciate the profound impact of this single publication, one must delve into the historical context, the experimental design, the immediate reception and lasting legacy of Hershey and Chase's groundbreaking. The intellectual groundwork for the Hershey-Chase experiment was laid much earlier in the 20th century. As of May 269, Friedrich Mercer first isolated what he called nuclein from the nuclei of pus cells. This substance, rich in phosphorus, was later found to contain DNA. However, for several decades, DNA was considered a relatively simple molecule, a monotonous polymer of repeating units. Proteins, with their enormous diversity of amino acids and complex three-dimensional structures, were favored as the likely candidates for carrying the genetic code. This protein paradigm dominated biological thought for much of the first half of the 20th century. Oswald Avery, Colin MacLeod, and MacLean McCarthy's 1944 experiment began to challenge this prevailing view. They demonstrated that DNA, purified from a virulent strain of pneumococcus bacteria, could transform a non-virulent strain into a virulent one. This suggested that DNA, in fact, carried the genetic information responsible for virulence. Avery, MacLeod, and McCarthy's work, though revolutionary, met with considerable skepticism. Many scientists found it difficult to accept that such a seemingly simple molecule could encode the complexity of life. There were also concerns about the purity of the DNA preparations used in their experiments. Some speculated that contaminating proteins, present in minuscule amounts, could have been the true transforming principle. Hershey and Chase entered this scientific arena with a keen interest in bacteriophages, viruses that infect bacteria. Bacteriophages offered an ideal model system for studying the fundamental mechanisms of heredity. These viruses are relatively simple, consisting of a DNA genome encased within a protein coat. When a bacteriophage infects a bacterial cell, it hijacks the host's cellular machinery to produce more phage particles. The crucial question was what component of the phage, the DNA or the protein, was responsible for directing this process. The genius of Hershey and Chase's experiment lay in its elegant design and execution. They employed radioactive isotopes to selectively label either the DNA or the protein components of the bacteriophage T2. They used radioactive phosphorus-32, denoted phosphorus-32, to label the DNA, as phosphorus is abundant in DNA but absent in most proteins. They used radioactive sulfur-35, denoted sulfur-35, to label the protein, as sulfur is present in proteins but absent in DNA. Next, they allowed the labeled phages to infect bacterial cells of Escherichia coli. After a brief period of infection, they subjected the mixture to agitation in a wearing blender. This shearing force detached the phage particles from the surface of the bacterial cells. The mixture was then centrifuged, separating the heavier bacterial cells from the lighter phage particles and the suspending medium. Hershey and Chase then measured the radioactivity in both the bacterial pellet and the supernatant. They found that a significant proportion of the phosphorus-32, denoted phosphorus-32 labeled DNA, was associated with the bacterial pellet, indicating that the phage DNA had entered the bacterial cells. In contrast, a large fraction of the sulfur-35, denoted sulfur-35 labeled protein, remained in the supernatant, suggesting that the phage protein coat largely stayed outside the bacterial cells. Furthermore, Hershey and Chase observed that the infected bacterial cells eventually lysed, releasing new phage particles. These progeny phages contained phosphorus but virtually no sulfur. This observation further solidified the conclusion that DNA, rather than protein, was the genetic material responsible for directing phage replication inside the bacterial cell. The Hershey-Chase experiment holds a pivotal place in the history of molecular biology due to its innovative methodology and unambiguous results. The use of radioactive labeling allowed for a clear distinction between the roles of DNA and protein during infection. The blender experiment provided a physical means of separating the phage particles from the bacterial cells, enabling a more precise analysis of their respective fates. Its methodological brilliance and clear results were undeniable. The acceptance of the Hershey-Chase experiment was far more immediate and widespread than that of the Avery MacLeod-McCarthy experiment. This difference in reception can be attributed to several factors. First, the Hershey-Chase experiment was performed using a simpler system, the bacteriophage, which was becoming increasingly popular as a model organism for genetic studies. Second, Hershey and Chase's experimental design was more rigorous, providing more compelling evidence for the role of DNA. Third, by 1952, the scientific community was becoming more receptive to the idea that DNA could be the genetic material. The growing body of evidence, including the work of Erwin Chargaff on the base composition of DNA and the emerging understanding of DNA structure, helped pave the way for the acceptance of Hershey and Chase's conclusions. The Hershey-Chase experiment had a profound impact on the future direction of biological research. It provided crucial support for the emerging field of molecular biology, which sought to understand biological phenomena in terms of the structure and function of molecules. The experiment helped to shift the focus of research from proteins to DNA, leading to a rapid explosion of discoveries about the structure, replication and function of DNA. The subsequent elucidation of the structure of DNA by James Watson and Francis Crick in 1953, just a year after the Hershey-Chase experiment, provided a molecular explanation for how DNA could encode and transmit genetic information. The double helix structure of DNA, with its complementary base pairing, provided a mechanism for accurate DNA replication and for the storage of vast amounts of genetic information. This discovery, building directly on the insights from Hershey and Chase, revolutionized our understanding of heredity. The implications of the Hershey-Chase experiment extended far beyond basic research. It had a significant impact on medicine, agriculture, and biotechnology. The understanding of DNA as the genetic material opened new avenues for developing diagnostic tests, therapies for genetic diseases, and methods for engineering organisms with desirable for example, the development of recombinant DNA technology, which allows for the isolation and manipulation of specific genes, would have been impossible without the knowledge that DNA is the carrier of genetic information. Recombinant DNA technology has enabled the production of life-saving drugs, such as insulin for diabetic, or has revolutionized agriculture through the development of genetically modified crops. The Hershey-Chase experiment also laid the foundation for the field of genomics, which involves the study of entire genomes. The sequencing of the human genome, completed in 2003, was a monumental achievement that depended directly on our understanding of DNA as the genetic material. Genomics has the potential to transform medicine by allowing for personalized treatments based on an individual's genetic makeup. While the Hershey-Chase experiment provided strong evidence for the role of DNA in bacteriophages, it did not immediately answer the question of whether DNA is the genetic material in all organisms. However, subsequent research confirmed that DNA is indeed the universal genetic material, with a few exceptions, such as some viruses that use ribonucleic acid, or RNA, as their genetic material. The impact of the Hershey-Chase experiment even extends to the realm of theoretical biology. The experiment's clear demonstration of the informational role of DNA has stimulated thinking about the nature of information itself. The concepts of genetic information, coding, and decoding have become central to our understanding of life and have even influenced the development of computer science and information theory. The legacy of Alfred Hershey and Martha Chase extends far beyond their single publication. Their experiment stands as a testament to the power of careful experimental design, rigorous execution, and clear interpretation of results. It exemplifies the scientific method at its finest and serves as an inspiration to scientists working in all fields. It is worth noting that the path to scientific discovery is seldom straightforward. The Hershey-Chase experiment, while ultimately successful, involved a great deal of trial and error. Hershey and Chase encountered numerous technical challenges in their experiments, including difficulties in achieving efficient radioactive labeling and in separating the phage particles from the bacterial cells. Their perseverance and dedication ultimately led to their breakthrough discovery. The relatively simple tools Hershey and Chase employed in their experiment highlight their ingenuity. This was long before the era of sophisticated molecular biology techniques. They primarily used radioactive isotopes, a Waring blender, and a centrifuge. The critical innovation centered on the method, using available tools for a novel experimental design to answer a fundamental question. The scientific community has recognized the significance of the Hershey-Chase experiment with numerous accolades. Alfred Hershey shared the Nobel Prize in Physiology or Medicine in 1969 with Max Delbruck and Salvador Luria for their discoveries concerning the replication mechanism and the genetic structure of viruses. While Martha Chase did receive less formal recognition during her lifetime, her contributions to the experiment were substantial, and her role is now recognized by historians of science. The Hershey-Chase experiment serves as a reminder that scientific progress often depends on the contributions of many individuals. While Hershey and Chase are credited with the experiment, they were part of a larger scientific community that included other researchers, technicians, and students who all played a role in advancing our understanding of genetics. In conclusion, the 1958 paper Independent Functions of Viral Protein and Nucleic Acid in Growth of Bacteriophage represents a pivotal moment in the history of molecular biology. The Hershey-Chase experiment provided compelling evidence that DNA, rather than protein, is the genetic material. This discovery had a profound impact on the future direction of biological research, paving the way for the elucidation of DNA structure, the development of recombinant DNA technology, and the rise of genomics. The legacy of Hershey and Chase continues to inspire scientists today, serving as a reminder of the power of elegant experimental design and the importance of challenging existing paradigms. The work stands as a testament to the transformative power of scientific curiosity and rigorous experimentation, a beacon illuminating the path to unraveling the mysteries of life.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.